Hello everyone. Today we are here to talk about the magical enzyme sucrase. Human sucrase, also known as sucrase isomaltase, is a digestive enzyme that catalyzes the hydrolysis of sucrose to its subunits fructose and glucose, which you then use for energy after eating. What about the function of sucrase? Sucrase actually has an incredibly important role during the final stage of carbohydrate digestion. This enzymatic process occurs on the brush border of the small intestine during digestion. Once sucrose is broken down into glucose and fructose, these sugars can be absorbed by intestinal cells and then transported into bloodstream for energy use by the body. So sucrase helps you eat and get energy. Invertase, a related enzyme, catalyzes a similar reaction, but in plants. Additionally, it happens to work on a broader range of substrates. While sucrase is specific to sucrose, invertase not only hydrolyzes the sucrose, but also other glycosidic bonds that contain fructose. Let's talk about sucrase down at the gene level. The protein coding gene responsible for giving the instructions for making the sucrase isomaltase enzyme is the SI gene. Sucrase is part of the glycosyl hydrolase family with a whooping length of 1,827 bases and a mass of 209,453 daltons. Sucrase also has 3 splice variants, all with similar lengths. Sucrase actually has 6 paralogs, which are genes that are related through gene duplication and are often quite similar in protein sequences. What about some additional structures that make sucrase unique to its function? Sucrase actually has 4 binding sites that can allow for substrate binding of sucrase and even 5 active sites for catalytic activity of sucrase interacting with its substrate occurs. Sucrase also has 5 disulfide bonds between position 63 and 646, contributing to the overall stability and strength of sucrase. All right, let's talk about post-translational modifications. Sucrase's precursor protein actually gets proteolytically cleaved when it is exposed to pancreatic proteases in the intestinal lumen. This removes the initiator methionine at position 1, which allows to activate the sucrase enzyme. Sucrase displays tyrosine sulfation at 7 different residues. Tyrosine sulfation essentially involves the addition of a sulfate group to hydroxyl group of a tyrosine residue in sucrase, which typically occurs in membrane proteins involved in cell addition, receptor signaling, or protein-protein interactions. Sucrase also displays phosphorylation at just 1 residue at position 7, where a phosphate group was added to sucrase, particularly by protein kinases on the side chain of specific amino acid residues like serine, threonine, or tyrosine. This process is very important for regulation of sucrase activity. Sucrase also displays glycosylation at a wide variety of residues where glycan chains are added to proteins. This process is actually quite important for the structure and function of these modified molecules. So what about the domains? Well, sucrase has 2 of them, P-type 1 and 2 domains. The P-type domains contain 6 cysteines linked by 3 disulfide bonds with connectivity 1 to 5, 2 to 4, and 3 to 6, which leads to a characteristic 3-leafed structure, the trefoil with 3 loop-like regions. The central core of the domain consists of short double-stranded anti-parallel beta sheet, which is capped by an irregular loop and forms a central hairpin at loop 3. The beta sheet is followed by a short alpha helix, with the majority of the remainder of the domain contained in two short loops which lie on either side of the central hairpin. Now let's talk about the exact mechanism of sucrase, which uses a hydrolysis mechanism for catalyzing products. The substrate sucrose, which is composed of bound glucose and fructose, comes in and binds to the two active sites on the sucrase enzyme. This interaction forms an enzyme-substrate complex. The binding of the substrate and enzyme puts stress on the glucose-fructose bond that make up sucrose and makes it eventually break. This breaking of the bond causes both glucose and fructose to be released from the enzyme, and this allows for the process to start all over again. In other words, sucrase catalyzes the hydrolysis of sucrose to produce glucose and fructose. Then the brush border transporters facilitate the absorption of these two monosaccharides. Now to our primary article. This article titled Blood Group Activity of Human Sucrase from Intestinal Metaplasia focuses on the role of sucrase in a physiological context outside of just digestion, helping us understand potential connections between sucrase activity, blood group interactions, and the development of intestinal metaplasia, potentially leading to stomach cancer, and whether sucrase may have something to do with it. So, what was the purpose of this study? Intestinal metaplasia is a condition involving the precancerous change of the mucosa of the stomach with intestinal epithelium and is linked to stomach cancer. Sucrase, an enzyme involved in digestion, is actually found to have variable ABO blood group activity, and it is sugar moiety, similar to cancer tissues. Blood group activity refers to the ability of sucrase to exhibit specific interactions with blood group substances, which are molecules present in our tissues that carry ABO blood group antigens. These ABO blood group antigens have specific sugar chains that determine a person's blood type. The study specifically compared sucrase in areas of intestinal metaplasia with that in normal small intestine with a particular interest in its ABO blood group activity in sucrase from areas of intestinal metaplasia. Now, why would sucrase have anything to do with cancer? Here is some evidence that may suggest a link. Intestinal metaplasia in humans resulting from chronic gastritis involves abnormal differentiation of stomach mucosa, leading to the emergence of intestinal-type epithelial cells. Enzymological studies have showed the presence of small intestine-specific enzymes in areas of intestinal metaplasia, particularly sucrase. Since stomach cancer often originates from these areas, research has focused on understanding its role in stomach carcinogenesis.
So let's talk about methods. How did they do it? To begin, tissue samples from gastric cancer patients were obtained and analyzed for intestinal metaplasia. Their ages, genders, and blood group types were recorded. Sections of the small intestine were obtained during gastric cancer operations from a patient, 61 female type B, and 2 colon cancer cases, 71 female type A and 56 female type O. The test tape method was employed to determine the area of intestinal metaplasia by isolating the mucous membrane from the submucosa, which was then stored at -70°C. Subsequently, sucrase purification was carried out, which involved solubilization with papain, ammonium sulfate fractionation, gel filtration on biogel P300, chromatography on DEAE cellulose, and electrophoresis on polyacrylamide gel to separate any contaminated minor blood group substances. Sucrase activity was measured using Dockfist's method with 0.1 molar sucrase as a substrate. Experiments on pH effects utilized a two-step method with a tris/glucose oxidase reagent and buffers of various pH values that all contained 0.1 molar sodium ions. For kinetic studies, 7 different substrate concentrations were used, and the Michaelis constants were determined by the Lineweaver and Burke method. Protein levels were determined by the Lowry method. Polyacrylamide gel electrophoresis was conducted using a 5% acrylamide separating gel. Gels were stained for protein with a Kumasi brilliant blue, or for sugar used the periodic acid shift method. Some gels were sectioned transversely, eluted in phosphate buffered saline overnight, and analyzed for sucrase and blood group activity. ABO blood group activity was measured using a hemagglutination inhibition test. The wells contained 25 microliter of antigen diluted with phosphate buffer saline. After 2 hours incubation at 37 degrees Celsius, 25 microliters of glutaraldehyde fixed red blood cells were added, and the inhibition titer endpoint was determined as a dilution inhibiting half of the hemagglutination. Now, let's move on to the results, starting with electrophoresis. The polyacrylamide gel was used for electrophoresis, and the results are quite interesting. Blood group activity was detected in 2 preparations of sucrase from the small intestine. The sucrase from a patient of blood type group A displayed hemagglutination inhibition of type A only, and sucrase from a patient of blood type group B displayed inhibition of type B only. The sucrase from a patient of type O, as expected, showed neither type A nor type B activity. Now, as for the blood group activities for the 5 enzyme preparations from areas of intestinal metaplasia, for those, preparations for 2 patients of type A blood showed no type A nor type B activity. One of the 3 preparations of type B showed neither type B nor A activity, whereas the other 2 preparations showed type B activity, but not type A activity. Here's the distributions of the 2 activities in the gels. The top one is from a patient of the small intestine group, type B. The other 4 are patients from the intestinal metaplasia group. You'll notice that the blood group activity was consistently found at the top of the gel. Minor protein and sugar bands at that position were also detected, possibly indicating the presence of glycoproteins or glycolipids interacting with sucrase. Interesting, right? Again, we see that only 2 of the intestinal metaplasia group had similar blood group activity as the small intestine group. So let's take a look at the results from kinetics and pH activity measurement. With sucrose as the substrate, the Michaelis constants for the purified enzymes from the small intestine and area of intestinal metaplasia were similar. Their Vmax values were also similar, being 25.6 units per milligram of protein and 23.2 units per milligram of protein, respectively. The pH activities of the 2 enzymes were also essentially the same, both peaking at a pH of 6. Despite the absence of blood group activity, as we mentioned earlier, the 3 intestinal metaplasia preparations exhibited similar enzymatic properties to sucrase from the small intestine. So, in summary of all that information we just told you, this is what it means. All patients of blood type A or B were found to secrete material with blood group activity. 2 of the 5 enzyme preparations from areas of intestinal metaplasia had similar level of blood group activity to those of preparations from the small intestine, and the other 3 preparations showed no blood group activity. Despite that fact, those 3 preparations exhibited similar enzymatic properties as the small intestine sucrase. Now, a possible explanation for the lack of blood group activity in these sucrase preparations could be attributed to incomplete sugar chains. Normally, ABO blood group antigenicity is determined by the terminal portion of its sugar chain, and it is normally present in several organs besides erythrocytes. Whereas recently, reports have shown that blood group activity is actually deleted in the stomach and colon tumors, and that new antigens that lack blood group sugar chains will appear in these tumors. Some changes of membrane glycolipids and glycoproteins have been associated with tumor formation, which could have been found to be due to incomplete sugar chains. Taking all that information into consideration, it could be a possible explanation for the lack of blood group activity. The study goes on to discuss that tumor-specific antigens have been found in gastric carcinoma, as well as areas of intestinal metaplasia. From this, researchers suggest that there may be a link between intestinal metaplasia and stomach cancer, and thus, sucrase might have a role in it. Overall, this study was able to explore the role of sucrase in a physiological context outside of just digestion, helping us understand potential connections between sucrase activity, blood group interactions, and the development of intestinal metaplasia, potentially leading to stomach cancer. Thank you all so much for watching, and don't forget to check out our references.